Good afternoon, everyone. I'm Kewalin. I'm a third-year PhD candidate at the University of Colorado Anschutz. And today I'm very excited to share with you the method I'm currently developing, SigMatch, a transcriptome-based regression method to detect mechanistic links across diseases and drugs. Let's start off with some background. Getting infection, we need two components, pathogens and the host. And most of the current treatments have been focused on using antibiotics to slow down the growth of the pathogen. Due to antibiotic resistance, unfortunately, those antibiotics not working so well anymore. And the question is, how can we help our body fight the infection better instead? The answer is yes, we can use host-directed therapeutics, or HDT for short, to help modulate the host response. So let's see some example of known HDT as adjunct therapy with antibiotics. Here we're looking at biological pathway involved in Mycobacterium tuberculosis infection in human macrophages. And our popular example of known HDT here are statins, which help us reduce lipid droplets in macrophages, resulting in inhibiting TB growth. However, discovering new HDT from scratch can take up to more than 10 years and very expensive. Computational drug repurposing accelerates this process by quantifying existing FDA-approved drugs for new drug indication. So what are the current drug repurposing out there? Given disease and drug differential gene expression profiles, we can perform pairwise comparison between individual disease and drug to identify a drug that best reverses the disease. Although they have shown lots of promise, these methods are not able to identify drug combination at scale. Another way is to use network-based method, which depends on predefined disease gene drug association. And unfortunately, when it comes to infectious diseases, which mostly understudy, we don't have this complete knowledge. And these two approaches we can identify two gaps. First, they only consider each disease in isolation and often overlook shared molecular mechanism across diseases. And this presents opportunities of leveraging what we already know from well-studied diseases to guide drug repurposing for less studied infectious disease. And this concept is known as transfer evidence and is already showing promise in practice. For instance, TB and COVID-19 share dysregulated type 1 interferon response and macrophage dysfunction. And TB HDT like metformin or statins are being investigated for COVID-19 due to their immunomodulatory effects. Therefore, we built SigMatch, a framework that learns mechanisms across diseases and drugs simultaneously from their differential gene expression and transfer the knowledge across diseases to identify drug opportunities in understudied infectious diseases. And we power SigMatch by integrating large-scale public dataset. We use process RNA-seq differential expression disease profiles from the SciAtlas database, which span over 600 diseases. For drug, we use drug profiles from links, including about 2,000 FDA-approved drugs profiles. And SigMatch was designed around the principle that effective drug combination should reverse distinct complementary components of a disease signatures, and this would maximize the therapeutic coverage. We begin with a disease target profiles and a set of drug profiles as references or features. Ideally, in the first iteration, SigMatch identified drug A, which reverses a portion of the disease profiles, and the next iteration select drug B to reverse the remaining unreversed component or error of the previous iteration. And then the next iteration, it would select another drug profile, for example, drug C, to reverse the remaining errors. And all of this it would provide us with comprehensive reversal. To achieve that, SigMatch used gradient boost GBM with monotone constraint. It approximates the first prediction by averaging feature values. And then at the next iteration, it would pick a feature to build a tree. And basically, it would correct the error from the previous iteration. And gradually improving the match between the input profiles and the combination of the feature profile at each next iteration. And altogether, this will be combined into an ensemble model that capture all of this complex relationship. And each of these features will be assigned importance score, which indicates how much the feature reduce the error at each iteration. And then in the end, we would compute a cumulative feature importance for each feature across all the iteration. So SigMatch can be used to identify two types of connection. First, direct connections identifying drugs for an infectious disease directly, and transfer connection, meaning that we can identify drug for, let's study infectious disease of interest through known drugs of better study non-infectious diseases. To illustrate how the potential of SigMatch, we perform an experiment called Leave One Out, where we start with features containing about 2,000 disease profiles covering 600 distinct diseases. We take one disease profile at a time and then use it as target, and we train SigMatch, then evaluate the disease profile associated with highest feature importance score. And the expectation here is that the chosen diseases are known to be mechanistically related to the target disease. For example, here, when we take our asthma, use it as target, the features that came up should be other asthma profiles and other lung-related conditions. And this heat map shows how SigMatch uses other diseases on the column to predict a target disease on the rows. And the darker blue here represent higher importance. And we can see that we have stronger signal along the diagonal, where target and reference diseases comes from the same system, which are endocrine, nervous, and respiratory system, suggesting shared biology. But do this important score actually reflect known biological relationship? And one way to examine that is through disease system, which are defined by shared anatomies or physiological function. And these systems are formally captured by biomedical ontologies like Mondo and DOID, which organize diseases into structure hierarchies based on shared ancestry. And this hierarchical structure of diseases let us compute semantic similarities, which tell us how closely related the two diseases are within the ontology based on their distance to a shared ancestor. For example, two respiratory diseases here, like asthma and COPD, will have a high similarities compared to a disease that's from a nervous system, like Parkinson and COPD will have a lower similarities. We use semantic similarity, specifically Resnick score from the Mondo ontology, to ask, does SigMatch prioritize biologically related diseases? And on this plot, we have higher importance score on the x-axis, aligned with higher semantic similarities on the y-axis, especially for diseases from the same system, which represent in the blue color. And this trend confirms that SigMatch capture real biological relationship. But what if same system diseases are not available? Can SigMatch still find relevant mechanism across system? And to test this, we remove all same system diseases from the features, forcing the model to rely on cross-system patterns. For example, if asthma is the target, all other respiratory diseases like COPD and TB in this case also will be removed And then this will show how well SigMatch can generalize beyond obvious biological similarities. As expected, removing same system diseases hurt the performance, especially for the target with strong signals, which show as the more number of regulated genes on the x-axis. And on the y-axis here, we show the performance, and you can see here that the performance is dropping. The orange here represent upregulated genes, and the blue one show downregulated genes. And this trend confirmed that SigMatch performance score reflect real biology, and that related diseases help the model learn even across less study conditions. Further, we also use TB as a case study to demonstrate SigMatch real-world potential. Even when we restricted to only blood-derived disease profiles, the model identified 55 diseases with positive feature important for TB. Many of these, like type 1 diabetes, mellitus, and Parkinson's disease, are also linked to drugs that's already studied for TB repurposing. And in conclusion, SigMatch employ gradient boosting regression with monotone constraint to enable scalable mechanism-guided drug repurposing for understudied infectious diseases. And our early experiment and result with about 600 diseases shows that SigMatch capture biologically meaningful signal among disease profiles. Also, the feature importance highlight mechanistically relevant diseases. And also the cross-disease learning reveal some known and novel therapeutics opportunities. And future direction, now that we have demonstrated the effectiveness of the transfer approach, definitely our next goal is to complete the other half of the framework to identify direct infectious disease drug connection. And the next phase includes tracing back the mechanistic path from the SigMatch top feature back to the target and trying to see if we can explain how the reference profiles mechanism collectively explain the target disease. Also, we would like to enhance the model even further by separating the shared tissue effects from the disease-specific signals. And more on the application for TB, we would like to train TB drug model to identify effective drug combination. And lastly, I would like to acknowledge both of my lab and both of my advisors, Dr. Jenny Ravi and Dr. Arjun Krishnan, and people in the lab, also the current and past funding for this project, GLBIO, for the opportunities to present my work today and for the travel fellowship. Thank you.